Hi, Miss Warren here, and I'm going to give you the directions for how to do the cell theory lab. All of the directions are written on this paper. They're taped at the instruction stations, they're at your tables, and they're at your lab station. So make sure, number one, that you read the directions. This is part one, animal cells. Only one of the two partners will do the animal cell slide. The other partner will do the pond water slide. Really important safety information. If you are sick, or have been sick, or think you're getting sick, you should not make the cheek cell slide because you could be spreading germs when you do it. Okay, so here's how you make the cheek cell slide. You're going to take a clean slide. Make sure it is very, very clean. If not, you want to wash it. Okay, and dry it with a paper towel and get it just super spotless, because if there's smudges on there, that's going to make it harder to see your cells. Your cells are very small. Okay, and then you are going to get a toothpick out of the box. And when you get the toothpick out of the box, don't reach in with your fingers because those have germs on them. You're going to reach in with a pair of forceps, not tweezers, forceps, and you're going to reach into the box and get out one toothpick. Once you have your own particular toothpick, you may hold it with your fingers. Okay. On your slide, see if I can do this. On your slide, you're going to put one drop of stain. This is a stain called methylene blue, and it will stain your hands. Not permanently, but your hands will be a little bit blue, so be very careful with it. It'll also stain your clothes. Okay, and you're going to put one nice drop, doesn't have to be very big, in the center of the slide. Then you're going to take the toothpick here, and you're going to rub it gently on the inside of your cheek. Do not use the pointy end, use the blunt end, and don't stab yourself. The goal is not to get blood cells here. You have cells on the inside of your cheek that are falling off all the time, and you're swallowing them and digesting them. They're falling off, so all you have to do to catch them is to gently rub and kind of roll the toothpick on the inside of your cheek. Looks like this. Again, gently, don't hurt yourself. Okay, then you're going to take that wet end of the toothpick and you're going to lay it flat in the slide, and you're going to smear it back and forth and kind of roll it. Okay, now this toothpick that has my saliva on it, my cheek cells, and stain on it, I'm not going to put that down on the table. I'm going to throw that directly into the trash can. The next step is to take my slide and put a cover slip on it to protect the lens from all that stain and stuff that's on the slide. So just like we did before, you're going to put the cover slip down at a 45-degree angle and drop it. Okay, you may see a lot of bubbles. I wouldn't worry too much about it. Just try to focus around the bubbles when you're looking at the slide under the microscope. Move it with your thumbs back and forth so that you can focus around the bubbles. You're looking for little blue specks. When you are done with both you and your partner looking at and drawing what you see on this slide, and there's some labeling you have to do, this slide you're not going to wash yourself because it could have germs on it, even though you don't think you're sick. So you're going to find the bleach box and you're going to put it into the bleach box so that it gets sterilized. Okay? And that's part one.